Good afternoon to you all. Respected chairperson, my dear delegates who are participated in from different parts of the world. So, I am Dr. Chandrashekhar. I am a professor of sericulture in the University of Agriculture Sciences, Bangalore, India. So, it is my immense pleasure to stand before you to present my research paper. So, my research topic, what I have done in our university is association analysis of major nutrient and secondary nutrient status of leaves of selected castor genotypes and growth indices of airy silkworm. So, we know we can the sericulture is mainly that is a culture of a silk. So, these can be classified based on the host plants, that is a mulberry sericulture and non-mulberry sericulture. So, mulberry sericulture which deals with the cultivation of silk from the mulberry silkworm, and non-mulberry sericulture it includes production of airy, tazar and moga silks. And based on the cultivation of host plants also we can classify agro-based and also forest-based. That is agro-based coming under mulberry, that is mulberry silkworm and airy silkworm, both are domesticated. So, we can grow this silk in domestic way, and if in forest based, those are tazar and moga silkworms. These are wild species, they can be reared in the forest. So, as we know, if we come to the global raw production, raw silk production as per the data of 2014, so the leading countries of the world which are involved in sericulture, silk producing are Brazil, Bulgaria, China, Colombia, Syria, Taiwan, Tunisia, Turkey, India, Indonesia, Iran, Japan, etc. So, China stands first in the production of silk, that is 1,46,000 metric tons, followed by India, that is 16% of total, that is 28,474 metric tons, which contributes about 16% of the total world production of silk. So, if we come to the silk map of India, so that is the major silk producing states we can classify that shown in the green shade, that is a mulberry silk, which is the India ranks second in mulberry silk production, is worldwide famous. So, next is the tazar silk. This tazar silk grown in area in Bihar, Jharkhand and Chhattisgarh districts of India, Chhattisgarh states of India. And then airy silk, it is a most famous in the states of Andhra Pradesh, Karnataka, and also some southern parts of southern states of India. So, this airy silkworm, we are going to deal with the study about the airy silkworm, that is a airy silk which spins a open type of cocoon, which silk is a non-reliable. It is also called haimsa silk. It is used in the spun silk industry. So, other one is a tazar silk, which is also wild species, which cannot be able to domestic. So, coming to the raw silk production in India, if we come to that mulberry silk, as I have told you already, mulberry silk contribute about 21,272 metric tons, which contributes nearly 75% of the total silk production among all the four silks are concerned. Then airy silk is 4,633 metric tons, which contributes 16.27% among all the mulberry silks and non-mulberry silks are concerned, followed by tazar moga. Moga, if you come to the moga, which is grown in only a state of Assam of India, which produces a golden yellow colored silk. So, India is having a credit, India is the only country which is producing all the four types of silk. Then this is a picture showing the image of mulberry sericulture. This silkworm, which is a monophagous, which feeds on the mulberry host and it spins the cocoons, and then later we can produce the finest silk, which is a worldwide famous. Then this is an image showing that is ericulture. As I have told you, this airy silkworm is a polyphagous in nature. It feeds on the primary, secondary and tertiary hosts, and then it spins a open type of cocoons. We can call that it as a haimsa silk. So, it produces that silk is a very famous in production of spun silk. Then this is a tussar silk. It is also a polyphagous in nature. It is a wild species, and it spins such type of cocoons, and the silk is also valuable. Then coming to the Muga culture, that is Muga silkworm. This is grown in Assam state of India. It is produces a golden yellow colored silk, which is a famous silk. It resembles a golden yellow type color, and it is a famous in India. India, of course, India is the only country which is producing all the 4 types of silks. Then coming to the host plants, as I have told you, we are dealing the Horticulture. If you come to the Horticulture, that is, it is eri silkworm is having the hosts of eri silkworm can be categorized into a primary hosts, secondary hosts, and tertiary hosts. So based on the preference of feeding, so we are categorized into a primary host, secondary hosts, and tertiary hosts. This primary castor, Ricinus communis, it is one of the primary host. If we put for example, if human beings, if we put so many type of different dishes, whatever we feel to eat, like we will prefer based on the preference of eating. So likewise, if we put all the hosts to the silkworm, this eri silkworm, it preferably feeds on the castor, that is Ricinus communis, and secondary hosts are Manihot and Evodia flaxisfolia. So tertiary hosts, Vithanthus excelsa, and other species. So as we come to the topic, that castor is the one of the ancient oilseed crop in the world. It is used for the production of oils. India accounts nearly 65% of the world's castor production. So in India, it's cultivated in about 6.28 lakh hectares in the states of Andhra Pradesh, Orissa, Gujarat, Karnataka, Kerala, Tamil Nadu, Maharashtra, Madhya Pradesh, Bihar, Rajasthan, Uttar Pradesh, Punjab, and West Bengal. So in Karnataka itself, that is castor is cultivated in the 24,900 hectares with a production and productivity of 25,989 metric tons and 1,095 kgs of productivity. So this, as we know, this tussar and muga, mainly they feed on the leaves. They are wild species, but preferably for the benefit of the farmer, the ericulture is a better to get the income. That I will come to the topic. The ericulture in 1 acre of land can fetch us minimum of 12 to 15,000 rupees per acre when entire foliage is used for eri silkworm rearing. So it is known that 25 to 40% of the foliage from the castor can be removed from the castor plantation. It can be utilized for eri silkworm rearing and without affecting the seed yield. That is the important thing I am going to stress. That is ericulture. Though castor can be used as both for the production of seed, and also apart from that we can use it for ericulture without affecting the seed yield. That is a dual purpose. We can say we can cultivate this ericulture for a dual purpose. So then there is in India there is a tremendous scope for ericulture in castor growing area without affecting the seed yield and seed production. It provides additional returns to the poor dry land cultivars and also small and marginal farmers. So castor is a minor oil seed crop, can be linked with ericulture to maximize the returns of the farmer. So with this introduction, the objective of this study is to study the major and secondary nutrients, because we have evaluated different hosts, primary host for this eri silkworm. That is to study actually the host varies with the quality. They are different in compositions, nutrient composition. In that context, we have taken this study to study the major and secondary nutrient status of castor in different genotypes, and to know the relationship between the foliar constituents of the leaves and also growth indices of eri silkworm. So in this we have taken this methodology, that is we have evaluated 8 elite castor genotypes. That is we have studied as per the package of practices and we have recorded the observations, the major and secondary nutrients of this mulberry leaf with a statistical analysis of RCBD, that is a randomized complete block design with the silkworm. We have taken agriculture also with a white plain silkworm breed, and with as per the recommended package of practices, and we have recorded the growth indices with a statistical analysis. So these are the varieties of the primary host, that is Erasinus communis. We have evaluated. These are varies with a morphological features also, apart from that foliar constituents in the mulberry leaf. So this is a DCS 9 variety, and that is then a 48-1 Kranti variety, DCH 177 variety. These are varieties, local and breeds, hybrids we are cultivating in different parts of India. Then these are also other varieties we have evaluated. That is DCH 4, DCS 32, DCS 85, and local varieties. Then we have recorded this foliar analysis of castor leaf. The leaf samples at 3 different heights of the plant. We have taken top, mid, and bottom, collected in different bags, and composite length samples were analyzed after 9 days of sowing of the seed. Then leaves were shade dried, and then with 3 replications. Then it is kept in the hot oven here of 70 degree until a constant weight is obtained, and the samples were analyzed. The major nutrients of the leaves were analyzed were nitrogen, phosphorus, and potassium. All this nitrogen, the estimated using 0.5 gram of sample. That is the procedure. Foliar nutrients were analyzed as per the Jackson's 1973. The same with the phosphorus analysis also, and potassium. And also apart from that, we have analyzed the leaf, the calcium and magnesium, and also a sulfur. These procedure we have followed as per the Jackson 1973. So we have recorded the growth indices of silkworm: larval weight index, larval duration index, pupal weight index, larval pupal duration index, cocoon weight index, silk index, and also apart from that eclosion index, oviposition index, a hatching index, leaf cocoon ratio, leaf egg ratio, leaf cocoon conversion rate, leaf silk conversion rate, growth index, that is a percentage of pupation and percentage of moth emergence, and net productive rate. All these observations were recorded after rearing of a silkworm. Then this statistical analysis is done as per the correlation coefficients were worked out to know the relationship between the foliar constituents and of the castor and growth indices is as per the Cochrane and Cox 2000. So the results of the experiments shown that the major nutrients of leaf versus growth indices of airy silkworm were the larval index were actually the significantly superior with respect to a larval weight index, pupal weight index, that is a cocoon weight index, with all the nitrogen, phosphorus, and potassium major nutrients are concerned. And then with major nutrients of leaf versus growth indices of silkworm were superior with respect to oviposition index, eclosion index, and hatching index, and also a leaf cocoon conversion. Whatever the growth parameters are concerned, it has shown significantly superior. So the same thing holds good for the minor nutrients, that is calcium, magnesium, and sulfur. So this is also secondary nutrients of castor leaves with growth indices also, they shown a superior results. So the outcome of this our research, as after evaluation of 8 castor genotype varieties, the results in a nutshell I wanted to tell, that is the foliar constituents of the castor genotypes like major and secondary nutrients had marginal variations with them, and even though they have shown significant variance. The growth indices of the silkworm also showed differences among various castor genotypes. So, and then the foliar constituents of castor genotypes could establish positive correlation with growth indices of airy silkworm. So, thank you for patient listening. If you have any questions, please
You have tried many castor genotypes.
Yes, sir.
Did you find that any silkworm has any preference to any varietal hybrid?
Yeah, that is a major. We have evaluated 8 castor genotypes. That is DCS 85 has shown superior response to this airy silkworm. That I am going to present in the same paper with respect to economic yield and also a preference of variety.
So why there is a preference to that hybrid?
Because there is, as I have told you, there is a variation in the foliar constituents. Suppose what we will take. So if we ate good quality of food, though definitely our health, growth and development and health and resistance will be good. Like that, there is a varietal variance in this there with respect to major and secondary nutrients. We found there is a variation in all the genotypes. Definitely DCS 85 and 48-1 found superior with respect to agriculture and also without affecting the seed yield. That's what I wanted to tell you in a nutshell.
Is there any question?
In agriculture, what is the additional expenditure that a farmer incurs?
So we have evaluated economics also, that I am going to tell you in the next economics. That is definitely without up to 50% of a foliage defoliation, we can work out without affecting to the seed yield up to a 50% of defoliation. That foliage can be used for agriculture. We can get a good revenue income out of that. Thank you.